There. What we're going to do today is talk a little bit about enzymes and what enzymes actually are. There was two ed puzzles, which hopefully, if you watch those first, this might make the note taking just a little bit easier. This is going to be on page 16 and actually 19 and 20 in your packet, so you can find those. There's some quick notes that go along with that. All right, so we're going to be basically looking at what an enzyme is. All right, so here's an enzyme. We're going to talk a little bit about that. Really, what an enzyme is a little molecular machine, all right? So an enzyme is really a molecular machine. And your body is full of these, and each of these little enzymes basically has a different job to do. It basically can make things happen. It forms things. It causes chemical reactions. So basically, we just think of them as tiny little machines that are in your body that are working all the time that help your body run. All right. A couple of things about enzymes. One is that they are what we call a catalyst. And what that is, it's basically something that speeds up a reaction. So there's lots of chemical reactions that have to happen in your body, right? And some of them would never happen if you didn't have a catalyst, or else they just wouldn't happen fast enough for you to survive. So these are really, really important. And that word is called their catalyst, right? Something that speeds up a reaction. The other thing to remember about enzymes is that they are reusable, which is really important in your body. So your body doesn't have to keep remaking these enzymes, which would take a lot of energy. It basically uses them, they do a job, and then they can be reused over and over and over again. So you can have a few enzymes and they can cause this chemical reaction to happen many, many times. All right. So that's another thing about enzymes is that they are reusable. The other thing is that they have a specific job. All right, enzymes can work on a specific, chemical reaction. And we often name them after that reaction they do. So here we might have lactase, which you might, if you are lactose intolerant, you know that you don't have that enzyme. Lactase acts on lactose. And it basically breaks down this complex or this enzyme, or actually the sugar, the sugar, and it breaks it down. So the lactase breaks this down into simpler sugars so that your body can use them, right? And if you don't have this lactase in your body, then you don't break down lactose, and you usually end up getting a sore stomach or other things. Your body just can't break it down and digest it properly. Okay. So three things. It's a catalyst, it's reusable, and it has a specific job. It works on a specific chemical reaction. All right. And here's a couple parts of an enzyme that are important. So here's my enzyme. And one thing to remember is that it's a protein. And we're going to look at that a little bit more in class. We're going to look at how those are formed. It's a protein. And we have, basically, there's something called the active site right here. All right. And it works on a specific substrate. So the substrate is what fits into the active site, in a sense, gets acted upon. All right. So here, when they're together, we call that the enzyme substrate complex. They fit together. And this is where the idea, and I'm going to put a little note here, is shape equals function. Okay. Often in biology, we talk about how shape matters. right? What it looks like and the way it's formed and what it looks like is really going to be really matter about how it works. Okay, so that's kind of something that's important to remember. All right. And you can see up here, we're looking at continuing, our substrate goes into this complex here, and then this one actually breaks it apart, right? So this is a reaction where these two chemicals are broken. So this might be an example of lactase breaking apart your lactose into two simpler parts, all right? That's something that our enzymes do. All right. Now, the way enzymes work, there's kind of two models. We're going to talk about the lock and key model and the induced fit model. All right. Lock and key model is that the substrate fits into the enzyme. It's a very specific shape. And only specific substrates will fit. So if I have lactose or lactase, which is the sugar, so let's say this is lactase or lactose, sorry. Lactose will only fit into this lactase enzyme. There's nothing else that can fit into there because it's a very, very specific shape. All right. So that's one way that enzymes work. The other is what we call an induced fit. And what happens is the substrate goes into the active site and that causes the active site to change shape. And this happens in some chemical reactions. We call it the enzyme hug. And there are some chemical reactions that this is how the enzyme works. It goes into that active site and the enzyme actually folds in around it and then causes the chemical reaction to happen. All right, so these are just two different ways that we're going to have enzymes work. One is called the lock and key. That's this first one here. So that's number one. And then the induced fit here is the second one. Okay, so two different ways that enzymes will work. All right, well, how does the environment affect an enzyme function? Right, enzymes are actually very, let's say delicate, but they have only certain ranges that they can survive. All right, so enzymes are basically, they're made of proteins. So enzymes are proteins. Oops. And they have a specific shape. All right. And then basically there's three factors you say that can mess with the enzyme shape. All right. And one is going to be temperature. And most enzymes are going to work at a certain range that's work best. If it's too hot or too cold, it won't work. So you're going to do a little activity where you'll kind of see a very familiar graph that kind of looks like that, right? And there's a range of temperatures. This was 0 and this is 100. there's a range of temperatures that are, I should put them down here, 0 to 100. There's a range of temperatures that that enzyme can work at, right? If it's too cold, it doesn't work very fast. If it's too hot, it doesn't work very well. It gets broken down. Just there's some range right here in the middle that it's the best. All right, so temperature is one. The second thing is pH. And that's basically how acidic or basic, so if it's an acid, like stomach acid, or a base, something can be very basic, which could be like oven cleaner if you have really strong base or really strong acid. And again, you would see that enzymes are going to have this this function that if it's very acidic or very basic, it won't work really well, but somewhere in the middle, it usually works the best. Now that can depend where you find it. So it depends on where in the body the enzyme is working. Right? It depends on the body where it is. So if it's something that's in your stomach, if you have like some kind of enzyme that's going to break down like meat, when you eat meat, you have pepsin, and that's going to break down that protein from your meat. It's going to have an optimal range would be over here. So it would have more of this kind of peak right here. And it would work the best at a very low acid, like maybe at 3. All right. But let's say you have another kind of enzyme, it may need something that's more basic. So these little graphs or these shapes are going to change depending on the enzyme. All right. And the third thing is basically salt concentrations. And again, it's going to have a certain optimal area that it works the best. All right. If it's lot salty enough, it may not work very well. And if it's too salty, it may not work very well. So again, a lot of these are going to have, basically, we're going to use the word optimum. They all have an optimum range, the best way that they work. And it may be at a specific temperature, at a specific temperature, at a specific pH, and a specific salt concentration. Right? So it's going to depend on the enzyme. Right? So they have certain factors in the environment that affect them. Now, enzymes also can be controlled. All right? We don't want enzymes to, they have a job and they have specific functions, but we don't want them to be working all the time. So let's say that you ate some milk, you drank some milk, and then you need this, your lactase to work. But when you're not drinking milk, you don't need lactase to work. So it only needs to be working when you need it. So your body's very good at controlling when things are working and when they're not. And then we're going to use something called inhibition. All right. Inhibition is a way to stop a reaction. So inhibition is basically turning off the enzyme. Right? And this can do this in a number of different ways. Sometimes we call this a non-competitive inhibitor. And what that means is that something is going to go into another place on the enzyme and cause the enzyme's shape to change. Okay, so this is a chemical attaches to the enzyme and changes its shape. All right, and of course, if I change the shape of the enzyme, I'm also going to end up changing the shape of the active site, and then the enzyme won't work anymore. Right, so that's one way that we can turn enzymes off and on, these little machines. So when we don't need them, we want to turn them off. Another way to turn off an enzyme is what we call competitive inhibition. Now, sometimes what this is that um, there is a chemical. I'm going to say that plugs up the active site. All right, so there's some way that we can basically plug up this active site over here with a chemical that sometimes looks very similar to our substrate, and it basically makes it so that enzyme can't work, right? So there can be something that has a very similar shape, and it can fill in that active site, and then the enzyme can't work. So this is going to be, it's a chemical that looks very similar to the substrate. All right. So that's just something that can control this enzyme. So sometimes we can change its effectiveness by temperature. We can also turn them off and on using basically what we call non-competitive, so something that's changing in the shape, and then something that can actually go into the active site. So this is just different ways that our enzymes can be regulated. All right, we're going to do a little activity with this, and you're also going to do a little online simulation with this so you can kind of see how that works. All right, and then we'll explore enzymes a little bit more when we look at some different factors in biology like photosynthesis, how your body works. So enzymes are just a really important protein that we're going to use and study.